Good morning, everyone. My name is Sakon Chankhachon. I'm a Ph.D. candidate in the data science program at Prince of Songkla University. Today, I will be presenting my dissertation research on the segmentation of retinal blood vessels in fundus images. Before I begin, I would like to acknowledge my advisor, Associate Professor Dr. Sathit Intachak, and Assistant Professor Dr. Supaporn Kansomkiat for their invaluable guidance and support through this research. My research title is Retinal Blood Vessel Segmentation in Fundus Images. Here is an outline of my presentation. I will start with the clinical motivation and problem statement, followed by a literature review and the identification of research gaps. Then I will detail my methodology, present the experiments and results, and finally discuss the findings and offer some concluding remarks. Let's begin with the clinical motivation and the problem this research addresses. Retinal image analysis is a critical tool in ophthalmology. Color fundus photography, or CFP, is a non-invasive method used to screen for major eye disease such as diabetic retinopathy, glaucoma, and hypertension. The primary goal of this research is to develop a fully automated system that can accurately identify key retinal landmarks. This will enable large-scale sprint programs, which are essential for preventing permanently vision loss. In the thesis, we focus on two key anatomical structures. First, the retinal vasculature, which is the network of blood vessels. Second, the fovea, a small depression in the retina that is responsible for our sharpest visions. Manually outlining these structures is a time-consuming and subjective process, which makes it a bottleneck for large-scale screening. Automated segmentation of these structures presents significant technical challenges. The blood vessels have clear linear features, while the fovea has very subtle and diffuse boundaries. This difference in features means that a model optimized for one structure may not perform well on the other. We can achieve accurate segmentation of both vessels and the fovea by using a two-pronged approach. First, by training our model on a comprehensively annotated multi-class dataset that provides the necessary anatomical context. And the second, by using a robust data augmentation strategy that can simulate the variation seen across different fundus cameras. There are several limit factors in retinal analysis. The first, publicly available datasets are often small, which can lead to overfitting problems. There is also significant variability between images from different clinical sites, a problem known as domain shift. Finally, inconsistencies in manual annotations can introduce noise into the data and degrade model performance. This research has three main objectives. The first is to construct a comprehensive multi-class anatomical data, a context-aware network for joint segmentation of the fovea and vessels. And the third is to develop a domain-robust augmentation pipeline. The corresponding contributions are the IDRID RETA fovea dataset, the MNV4 fovea model, and a novel GV-based histogram augmentation techniques. Now, I will provide a brief overview of the existing literature and identify the research gaps that this research aims to fill. The retina contains several key structures that are important for diagnosing eye disease. The macula and fovea pit are crucial for high acuity vision, and the foveal avascular zone, or FAZ, is a key biomarker. Pathologies associated with diabetic retinopathy, such as microaneurysms and hemorrhages, often appear alongside changes in the vessel structures. This slide shows some examples of common lesions found in the patients with diabetic retinopathy. Microaneurysms are the tiny red dots and are earliest sign of disease. Hemorrhages are larger, bottle red spots. Heart exudates are yellow deposits that indicate vascular leakage. And soft exudates or cotton wool spots are called fibrillary lesions that indicate nerve damage. For the purpose of this thesis, we only be focusing on the anatomical structure and not this region. The field of retinal segmentation have evolved significantly. Traditional methods for vessel segmentation rely on the techniques like match filters and risk tracking. But these methods require manual tuning and were not robust to pathologies. Deep learning approaches, particularly UNet and its variants, have become the new standard. For fovea localization, early methods used anatomical relationships and intensities, but these failed when the anatomy was disrupted by disease. More recent deep learning methods use and diffusion models for synthesizing realistic data. Traditional methods for vessels treat vessels as rich matched filters and Hessian filters were used to create vessel probability maps. Risk tracking algorithms follow the path of the vessels. Wavelet and Garber scheme were used to capture variance in appearance, and mathematical morphology was used to extract the center line of the vessel. However, all of these methods had limitations, including the need for manual parameter tuning and lack of robustness to pathology and image quality variations. Modern deep learning approaches have largely overcome these limitations. UNet and its variants are strong baseline for this task. Attention mechanisms have been incorporated to help model focus on the vessel-like patterns. The current frontier in this area is the use of transformer-based models, which can integrate global context more efficiently. The key trends in this area are the development of lightweight models for deployments, richer feature fusions, and the incorporation of geometric prior to improve robustness. Traditional methods for Fourier localization rely on the Fourier low intensity and its geometric relationship to the outer landmarks, such as optic disc and major bud cells. Some methods also leverage the parabolic trajectory of bud cells or inherent symmetry of retinal structure. However, these methods often fail when the normal anatomy was disrupted by the pathologies. Contemporary deep learning approaches have proven to be much more robust. For example, heat map regression, where the model predicts a probability map of the Fourier location, is a common technique. Multi-task learning, where the model is trained to simulate author structures, has also shown to be effective. The most recent and successful approaches use transformer-based models to fill global contexts with local features. The trend in this area is move away from the simple regression and towards more complex context awareness models. In this slide, it's about data augmentation. Data augmentation is crucial in medical image due to a small size of datasets and class imbalance. Conventional augmentation techniques include geometric and photometric transforms. More advanced methods like mix-up and cut-mix blend images or patches to regularize the models. The current frontier is synthetic data augmentation using GANs and more recently diffusion models, which can create highly realistic labeling data to reduce the burden of manual augmentations. In summary, the priori works in Wassel and Fourier segmentation has progressed from classical filter-based methods to modern deep learning approaches. The current research frontier, which my work at Thesis, is focused on improving generalization and fusing global and local contexts to overcome the limitation of earlier models. Now, I will describe the methodology used in this thesis. Our methodology consists of 5 main steps. First, data construction, pre-processing and augmentations, network architecture design, training, and evaluation. This diagram provides a high-level overview of our pilot. We start by constructing the IDR-ID Rita Fourier dataset by merging and manually annotate data from IDR-ID and Rita datasets. We then apply our GV-based histogram specification for data augmentations. The augmented data is then used to train our MNV4 Fourier models, which is based on a MobileNet V4 backbone and a deep lab. A segment image with labels for the Fourier, optic disc, retina, cells, and background. Our first contribution is the IDR-ID Rita Fourier dataset. We created this dataset because existing datasets lack the comprehensive multi-class annotations needed for contextual learning. We merged annotation from IDR-ID and Rita datasets, manually designed the Fourier, and has all labels verified by a senior ophthalmologists. The final product is a 5-class dataset that enables a model to learn the critical spatial relationship between retina structures. Here is a sample image from our proposed dataset showing the original Fundus image and the corresponding multi-class annotation. Our second contribution is a domain robust augmentation pipeline to powered by different Fundus cameras. We use a technique called histogram specifications. We model the color profiles from external datasets use a generalized extreme value. Our RGB distribution then transfer these profiles to our training images. This expanded our training set from 54 to over 2,000 high-quality images and forced the model to learn features that are invariant to color and brightness variations. Our third contribution is the MNV4 Fourier architecture. This model is built on the efficient Deep Lab V3 architecture and pre-trained MobileNet V4 encoder. We specifically modify the architecture to better capture and fill features at different semantic levels, which is critical for Fourier localization. The model processes information at 3 distinct levels. Low level for basic identify the Fourier. This visualization shows how the model learns. In the low-level features, you can see that the initial layer identifies basic edges and color gradients. In the mid-level features, the model starts to assemble structure components like the Wassel network and the optic disc. The approximate location of Fourier begins to emerge. In the high-level semantic features, a strong localized activation appears at the Fourier, indicating that the model has successfully integrated global spatial context. Finally, in the decode output, these hierarchical features are filled to produce the final precise multi-class segmentation map. For training, we use the PyTorch framework on a system with 4 NVIDIA RTX A4000 GPUs. We use the Adam optimizer with a batch of size 16 and a multi-class cross entropy loss function. The model was trained for 1,000 epochs with a learning rate of 1e-3. For evaluation, we use comprehensive set of standard metrics, including intersection over union or IOU, recall, precision, F1 score or dice, and accuracy. Now, let's move on the experiments and results. Our model was trained exclusively on our augmented ID Rita Fourier dataset. We then test its generalized performance on a comprehensive set of a public dataset with a detailed in table 4.1 and 4.2 of the thesis. We evaluate segmentation quality using IOU and F1 score, and localization accuracy using average Euclidean distance AED, and success detection rate, or SDR. To validate our hypothesis about the importance of anatomical context, we conduct an ablation study. We train 3 models with increasing level of anatomical information. The first model trained only on the fovea and optic disc completely failed to localize the fovea. When we added the retina as a contextual clue, performance improved dramatically. Finally, with the full context, include the vessels, the model achieves strong and accurate performance. This definitively proves that providing multi-class anatomical context is essential for accurate fovea localization, improving recall from less than 1% to nearly 90%. Our MNV4 fovea model established a new state-of-the-art in fovea localization accuracies as measured by the average Euclidean distance across 3 challenge public datasets. Our model consistently achieved the lowest AED, significantly outperforming prior state-of-the-art methods. The success detection rate, or localized accuracy, it measures the percent of test images where the predict fovea center lies within a specific distance from the ground truth. The distance is normalized by radian of the patient's optic disc to account for the variation in scale between different eyes. Our model demonstrates excellent performance on the SDR matrix. On the Mercier dataset, it outperformed other methods at the finite localization threshold and achieved near perfect detection border radians. On the Refuji dataset, the SDR was lower than 5 scales, but we attribute this to inaccuracies in the ground truth, which I will discuss later. Overall, the model demonstrates highly reliable detection rates, particularly on accurately labeled datasets. Our framework also achieved robust and generalization performance for vessel segmentation. The models demonstrate strong cross dataset generalization, achieving testing both major vessels and find capillaries with minimal false positives. Qualitative analysis further confirmed the model's robustness. In challenge cases, the confounding lesion, the model accuracy identified the fovea by leverage global context. We also observed the impact of labeling inconsistencies. On the Maple-DR dataset, a discrepancy between the small ground truth fovea label and our model's more anatomically pleasing prediction led to poor quantitative scores. Despite a visual correct segmentation, this highlights how inconsistent labeling conventions can distort quantitative metrics. Now, I will discuss the implications of our finding and offer some concluding remarks. One of the key takeaways from our of annotation qualities. On the Refuji datasets, we observed that our model performance seemed lower by some metrics. Upon closer inspection, we found a systematic discrepancy between the model's ground truth and actual foveal location as verified by an expert. Our model prediction was actually more accurate than the provided ground truth. After re-annotating a subset of data, our model F1 score improved from 0.68 to 0.98. This demonstrates that the model performance on public dataset can be limited by the quality of the ground truth data itself. In summary, we demonstrate that anatomical context is the most critical factor for robust segment low contrast structure like the foveals. Our proposed CEV-based augmentation strategy effectively overcomes domain shift, enabling superior generalization on unseen datasets. This research has developed a robust context-aware framework that significantly advances the state-of-the-art in the retinal image segmentation. For future works, we plan to integrate the model into end-to-end system for automate diabetic retinopathy gradings. We also plan to expand our training dataset with more examples of diverse and rare pathology to further improve robustness. Finally, we will explore model compression and quantization to enable deployment on low-power mobile devices for use in primary care and underserved areas. The work presented here has been submitted to IEEE Access and is currently under review. Thank you for attention, and now I'm happy to answer any question you may have.